Okay, by the end of today, I want you to be able to, this was yesterday's learning outcomes, not today's. But remind yourself, yeah, we're more at the bottom here. So in general, from these yesterday or Wednesdays and today's lectures, you should be able to list the hormones secreted by the pancreas and the cell types making up the pancreas. Just remember, cover all the biogenesis of the peptide hormones that we talked about. Describe the process of insulin secretion, its mechanism, the synthesis of action and function of glucagon. Remember the differences between peptide and steroid hormone cells. Think about gene duplication and those four mechanisms of how hormone diversity is achieved. Then we're going to talk about specifically today this mechanism of glucose-induced insulin secretion, regulation of insulin secretion, mechanisms of action and biological functions of insulin, and some of the physiology and biochemistry of glucagon. We'll also briefly cover some of the other islet cell hormones, because remember there's multiple islet types within the pancreas. So we'll start here because we left off on peptide hormone diversity. So let's discuss exactly how glucose-induced insulin secretion occurs. And remember that the beta cell is a peptide-optimized cell type. So it's got lots of rough ER. It's got plenty of mitochondria to create ATP and energy for the system. It has a large endocytic system with lots of vesicles, and is thus easily recognized. Okay, so this is a very, 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 very simple sketch of a beta cell, and you would have glucose on the outside that would then enter into the cell via GLUT2, or in general high affinity glucose transporters. This first step is dependent on the enzyme glucokinase, which is the first step of glucose metabolism. Glucose is metabolized by this glucokinase. Then the metabolism of glucose results in an increase in intracellular ATP. This is a reminder from all your biochemistry pathways that you learned about glucose, what happens when glucose is taken up. And obviously, thus the importance of all the mitochondria located within the cell, because a lot of this biochemistry occurs within those mitochondria. So here you can think about all those enzymes in the process for breaking down glucose and driving ATP. Your hexokinases. Then, once ATP is produced, increasing levels of ATP will cause the closure of an ATP-sensitive potassium channel. Now, what do you think this is going to do? Well, typically, or oftentimes, potassium is linked with other channels, and indeed what this does is it drives depolarization of the membrane and opens a voltage-gated calcium channel. This then brings in calcium from the outside and starts to drive mobilization of preformed insulin secretory granules within the beta cell cytoplasm. And this is due in large part to calcium-sensitive proteins, which are responsible for vesicle trafficking and vesicle fusion and vesicle formation. Insulin then gets released as vesicles fuse with the plasma membrane. Now, there are two very important signals within this process that I've just talked about, and those are the increase in ATP due to the breakdown of glucose and the influx of calcium from the extracellular via the opening and activation of that calcium channel due to depolarization of the membrane. So this leads these two different key steps are set up what is a two-phased process of insulin secretion. The first phase is very rapid. Okay, the vesicles are already docked at the plasma membrane, roughly 5% of them, and they're ready to quickly release into the bloodstream upon a trigger. The second phase is prolonged secretion, and this requires processing of proinsulin, which requires much greater movement of vesicles. So this is about 95% of secreted insulin. And remember, from what we talked about yesterday, it takes time to make proinsulin and to process it, okay? You've got to have all those cleavage events. You've got to move it through the endocytic system, through the Golgi, and to form the final insulin peptide. So this prolonged step has a higher threshold for when it's going to get secreted. The cell wants to really make sure that insulin needs to be secreted before it embarks upon or starts a process of making more and more insulin, because insulin takes energy, it takes resources, and it takes time. So calcium has two major effects on the control of insulin secretion. The first is to promote secretion of these preformed secretory granules. It's the first phase of insulin release. It's that 5%. Secondly, calcium is required for those peptidases. You remember the PC2 and PC3? Those endopeptidases, which have activity, excuse me, within the secretory granules. And this activity is sensitive to calcium levels and are what do the processing of proinsulin to insulin. So in the first phase, calcium impacts calmodulin, it impacts some of your RABs, some of your COPs, some of your clathrin-coated pits, all the things that lead to trafficking and release and fusion of the vesicles. And in the second phase, calcium is required for this activity of these endopeptidases that are driving and setting up and making insulin. For ATP, it has also two major effects on the control of insulin secretion. It depolarizes the membrane by closing potassium channels, which then leads to an influx of calcium, which obviously impacts the first phase of insulin release. And in the second, it provides an energy source to move those vesicles along the endocytic trafficking system, which obviously includes microtubule-associated motor protein, or kinesin, which is responsible for moving these secretory granules to the plasma membrane. And this is kinesin requires ATP to function. So it uses ATP to move along and step along these microtubules, which help drive the vesicles eventually to the plasma membrane to prepare for the second phase insulin release. So what does this kinesin-dependent movement of secretory granules look like? In the first phase, you have kinesin that is walking up. You remember that microtubules have a negative and a positive direction, and they grow and shrink in these two different directions. In red here, I'm showing you this secretory granules. The kinesin is made up of heavy chain and light chain. The heavy chain has what looks like, almost walks like a penguin. And it alternates feet on the microtubule. The light chain then binds to the secretory granules via adapter proteins. Then once you move up this microtubule, the microtubules then drive and point towards the plasma membrane, and this sets up for the second step. So you've got this walking secretory granules bound to the light chain, moving up the microtubule towards the plasma membrane. And this is, if you remember that image I showed you of a beta cell with all the little black circles and dots of vesicles, this is driven by kinesin and microtubule movement, moving up and sitting there right at the edge of the plasma membrane, ready for that first wave of calcium. And sorry, I should say that it's important to remember that this is, they're sitting there for the first wave, but this process then recycles itself for the second wave, okay? Because you can imagine to go from all the way from the Golgi to the plasma membrane takes a lot of ATP. So the cell really wants to commit itself and recognize, okay, is this really something we want to do? Do we want to move all this insulin to the plasma membrane and release it? Because it's going to take a lot of energy and resources. So kinesin, there was a lot of studies when I was in graduate school about the mechanism of kinesin and how it worked, and purifying it and studying it. And I remember when some of the first movies came out,
the cell, kinesins pull organelles along microtubule tract. The organelle attaches to the other end of the long frail coil that holds the two motor heads together. The organelle is not shown here. In solution, both kinesin heads contain tightly bound ATP and move randomly, driven by granular motion. When one of the two kinesin heads encounters a microtubule, it binds tightly. Microtubule binding causes ATP to be released from the attached head. ATP then rapidly enters the empty nucleotide binding site. This nucleotide exchange triggers the nectin to zipper onto the catalytic core. This action throws the second head forward and brings it near the next binding site on the microtubule. The attached trailing head hydrolyzes ATP and releases phosphate. As the neck linker unzippers from the trailing head, the leading head exchanges its nucleotide and zippers its neck linker onto the catalytic core, and the cycle repeats. In this way, kinesin dimers move processively, step by step, along the microtubule.
So one of the things that strikes me is just look at all the ATP. I'll just play that last part. Look at all the ATP required just to make one or two steps, right? So to take hundreds of vesicles to the plasma membrane along microtubules, think about the amount of ATP that is required to do that. Okay? And that's what I want you to understand and appreciate, that the amount of energy for the second phase, well, just even for the first phase, to get those vesicles to the plasma membrane is tremendous. So for the cell to embark on the second phase, it really wants to make sure that that's what it wants to do, and trigger and drive, use ATP and release insulin. So there are a lot of mechanisms, and that's why it's so important to have a threshold of signal to control whether or not this process is starting and embarking. Now, just to summarize, when glucose binds to the GLUT transporter and enters, two very important intracellular changes occur which lead to insulin secretion. First, you have calcium, you have depolarization of the membrane. Well, first you have metabolism of glucose, release of potassium outside of the cell, depolarization of the membrane, and then intracellular movement of calcium from the outside to the inside. And this then mobilizes insulin secretory granules in that first phase, so roughly 5% of the vesicles, and then eventually triggers activation of the endopeptidases, which start to allow processing of insulin from proinsulin and to facilitate the second wave. In combination with this calcium, you have increased production of ATP because of your breaking down and metabolizing glucose. This will then close the potassium channels and driving those that depolarization event for the first phase of secretion, and eventually then promotes vesicle movement by providing an energy source for kinesin to then drive that second phase of vesicle movement. So we've covered now regulation of insulin secretion. And can you just give me, apologies guys, something's happened in my house. I might need to run, take care of it for a bit. We might do an early pause. Can we take a pause for half an hour, and then I come back and finish this second half. The second half will be relatively short, but there's something in my house that needs urgent attention. I apologize. Okay, so we take a 30-minute break, and then I come back in 30 minutes and we'll finish it off. Okay? Apologies.